of mine is looking for some help in the lab. She works on fly olfaction, pretty cool stuff. If you're interested in that position, hopefully you can see that contact information, but I'll make sure that this slide is in the lecture slides that I posted for today. And you have to promise to do a good job because she's a good friend of mine. So if you don't, I'll know. So if you're interested in that job, go ahead and contact her, and she can give you more details about it. Okay? All right, so today we're going to continue with mitosis and begin to get into DNA synthesis and replication, which is the last topic before the exam. Just a reminder, so that no one can say that they didn't get the information. The exam 1 review session is on Tuesday here from 5:30 to 6:30. I do think, so you're not going to like this next bit, I'm going to give you a very short homework assignment tonight to make you look at the material we're going to be covering today and Monday. So you don't wait to the last minute to start looking at that material, the last bit of material for the exam. So basically I'm going to give you 1, maybe 2 questions on DNA replication, just to get you start working with the material before we cover it in lecture on Monday, so that it's not completely new, and you have some of that information to begin working with over the weekend. Okay? So look for that. I'll post it after class today. And that will be due on Monday, right? Short 1 question. It's not too bad. It's related to the exam. All right, so very short homework assignment for this weekend. All right, any questions before we get started with lecture, the exams, the homework? One other thing, as far as just class logistics and record keeping after the first exam, once we get those grades done, just so you know, it'll probably take me about a week with the help of the TI to get all the exams graded. We'll do it as quickly as possible. It'll probably be the following week that you get those grades back. We'll post them in Lalima, and when I post those exam grades, I'll also post all the current grades I have for you as far as homeworks, quizzes, and participation points. So you'll get a chance then to check in, make sure everything's been recorded properly, making sure that your clicker is working, okay? So I'll post all those points at the same time after the exam once we get those graded. All right, and I just want to, a few of you have emailed about clicker issues. I just want to remind you that you don't need to purchase the repolling access, that account. You need to be registered in it for me to track you, but you don't need to purchase it if you just have a clicker device. Okay? All right. So this is where we left off on Wednesday, talking about, we made it all the way through anaphase and mitosis. We're talking in detail about the forces that are driving the cells apart and pulling the sister chromatids apart in that phase. So we were talking a lot about microtubules, microtubule structure, and then the motors associated with those microtubules, and specifically how they're acting during mitosis and anaphase to achieve these movements. All right, so today we're going to go over in detail the 3 types of motors and movements that I expect you to be familiar with and exactly how those work. We talked a lot about motors and how they move microtubules around in previous lectures. All right, so hopefully this will be fairly straightforward for you. So we'll go through each one of these types of motors and how they're acting in mitosis one by one. All right, so the first one, we look at the polar microtubules. These are microtubules that, rather than connecting to the chromosomes, they interact with another microtubule. And that interaction is through a motor protein. And this motor protein is helping to drive the cells apart. All right, so if you look at that schematic, the polar microtubules and the motor that is interacting between those microtubules. All right, if it's moving, well, cells apart, right? They're interacting with microtubules, not directly with the chromosomes, but that's also helping to force the chromosomes apart. All right, so what kind of motor must it be? The motor between two microtubules that's pushing the cells apart. That plus or minus end directed. Remember all the poorly put together animations I showed you of the little stick man moving microtubules around? So it's actually, that should be microtubules are moving the cell apart, which is helping to pull those chromosomes apart, but it's an indirect thing. So that should say cell. Right. The motor acts at polar microtubules to move cells apart, which thereby helps move those chromosomes apart. What kind of motor must it be? We're talking about that motor between two microtubules, two polar microtubules. All right, think about what way microtubules are being moved. Think of it as a force that is pushing the ends of the cells apart. Alright, has everyone had a chance to answer? There's last minute answers in. Let's take a look. All right, a little bit of confusion. But it is indeed a plus end directed motor. All right, so let's look at why that is. All right, remember this guy? If we are pushing apart the ends of the cells, right, that means the microtubules have to be forced in that direction, away from each other, which means that that motor between those two cells, or two microtubules, is a plus end directed motor. To go back. All right, so this schematic. Microtubules are moving away from each other, which means those motors have to be pulling towards that positive end. Which means they are what kind of motor? The kinesin. Kinesin is our positive end directed motor. All right, let's look at the astromicrotubules. So these microtubules that project from the centrosome towards the edge of the cell, and they're anchored into that cell membrane. All right, so we have that motor as that anchor point. So in order for this motor to help in pulling apart the cells and pulling apart those chromosomes, so take a look at that schematic. So a motor acts at the membrane to impact those astral microtubules and move things apart. And what kind of motor must it be? Anyone want to see the schematic again? Yes? All right, what kind of motor must those be? Those red inchworm looking things. All right, everyone had a chance to answer? Take a look. All right, still a little bit of confusion, but the majority of you got that right. It's minus indirected. All right, if we look at this schematic, in order for that to be helping to pull that spindle apparatus apart, it has to be moving its way towards, pulling that centrosome towards it, right? Which means it's moving towards the negative end. All right. So the final movement we're going to look at is that kinetochore, that bit that's attached to the chromosome and is actually helping, or the forces that are pulling apart those sister chromatids. So we're going to look at these motors, and unlike polar microtubule motors and the Astral motors that we just looked at, Astral motors, what kind of motors are they then? Dynein. All right. So unlike those two where they're examples of motors that are common in the cell, doing the same sort of thing that they do other places in the cell, the kinetochore is a much more complex machine. So I think that I have a video here to help illustrate some of that, about what's going on with the kinetochore during anaphase. Maybe. Or not. Right, well then. So you don't have to go watch that on your own, because it's not going to play today. Basically, the first part of this shows chromosomes and how they're wrapped around histones and then packaged up, that we've already talked about, into coils, and then the coils into loops, and then those loops packaged together. And then the second half talks about the kinetochore and some of the machinery involved in that. All right, but I have some slides to show you in a less interesting way than the video. All right. So just a general question, an Dynein mutant, how would you expect the chromosomes to behave at anaphase? Where have we seen Dynein acting so far? That's the Astral. So if we mutate that Dynein, what would happen? You guys are quiet today. Mumbling. All right. So what do the Astral motors help do?
Separate the cells.
Thank you. Separate the cells. All right. There are other forces helping to push that apart at the polar microtubules, but that's going to slow down that process. We're going to look at how Dynein also plays a role in the kinetochore. All right. So it's also going to affect the pulling apart of the sister chromatids. That is what the kinetochore looks like. It's an incredibly complex set of proteins that are involved in hanging on to that chromosome at the centromere, hanging on to the microtubule, and then also there are a lot of proteins there for sensing the tension. Right? They don't start pulling apart until the tension is just right. So there are a lot of proteins there. I don't expect you to know those all. A few to point out that are of interest to us that we've already talked a little bit about. That CNPA, that's a modified histone protein. That helps to hold, for that kinetochore molecular machine to hold on to the chromosome. All right, so a similar function to the histones we've already looked at. It's interacting with the DNA, and it's helping to hold that kinetochore onto that structure. CNPE, that's a modified kinesin. So that helps also anchor the microtubule to the chromosome. And then also notice we have Dynein here. All right, so if we take, these are just the proteins involved, we can look at a little bit more closely at the structure. So that CNPE, that modified kinesin, that helps hang on to the end of the microtubule and anchor that microtubule to the chromosome. Right, by interacting with the kinetochore, which is anchored to the chromosome by that modified histone. And you can see that there are a lot of proteins involved in anchoring that microtubule onto the kinetochore. Remember that microtubules have that dynamic instability. They're always growing and shrinking, so it takes a lot of anchoring to hang on. Right? Part of that is a modified kinesin. It's still plus N directed, doesn't have much plus N to go there. So it's just working on holding things together. So you have the action of both this modified kinesin and then a dynein, which is negative N directive, to pull things apart once, but they only do that once they get the signal that the tension in that microtubule structure and that spindle apparatus is correct, once everything is lined up. All right, so they're working together to anchor it and then pull it apart. So if we look at these together, all right, so we have at the minus end, depolymerization, which helps shrink that microtubule from that end. Right, so we have forces at both ends that are helping to contract and pull those chromosomes apart, those sister chromatids. So at one end you have depolymerization, so you have subunits falling off to shorten that microtubule. At the other end you have that modified kinesin anchoring it in, and once it gets the signal, that dynein starts walking down the microtubule and pulling everything with it. All right, so it's a very complex machine. It involves lots of proteins, including 3 we've already talked about, that modified histone for hanging onto the chromosome, that modified kinesin for hanging onto, anchoring the plus end of the microtubule to the kinetochore, and then that dynein to pull everything down once it gets the proper signal. All right, so there is what you need to know as far as motors and microtubules and the forces driving the cells apart and the chromosomes apart during anaphase. 3 types of microtubules, 3 types of motors, and whether they're positive or negative N directed. All of these work together in anaphase for that anaphase 1, separation of the chromatids, and anaphase 2, separation of the cells. All right, so what happens to chromosomes during mitosis? You had to sum it up in one sentence. Replication, separation. Anybody else have a one-sentence summary of mitosis?
Pulled apart.
Here's something in the back.
Pulled apart.
What's pulled apart?
Chromosome, the sister chromatids.
The sister chromatids are pulled apart. Right, that's the whole goal of setting this all up. Separation of those sister chromatids. That is the goal of mitosis. All right. So quick summary of mitosis before we move on to replication. We have interphase, where we have the DNA replicated, as well as the centrosome, 2 centrosomes, 2 copies of all the chromosomes that are still associated as sister chromatids. Prophase, those centrosomes start to move apart. You get the beginning of the formation of the mitotic spindle. You still have that nuclear membrane, but the DNA is condensed. Prometaphase, nuclear envelope disappears. You begin to get attachment of the microtubules to the chromosomes at the kinetochore. Centrosomes have moved to the poles. Metaphase, the spindle apparatus has pulled on those chromosomes and lined them up at the equator, metaphase plate. Anaphase, we get separation of those sister chromatids, and the ends of the cells are beginning to be pulled and pushed apart. Finally, the last two stages that we haven't talked about yet, telophase and cytokinesis, where you get the chromosomes decondensing, you get the reformation of the nuclear envelope, and then you get cleavage furrow, and finally division of those cells. Now, oftentimes, when you get to this point in mitosis, people just say telophase, cytokinesis. It happens sort of together. But I want to make the point that they are actually referring to two separate things. Telophase has to do with what's going on with the chromosomes. Cytokinesis has to do with what's going on with the cytoplasm and the cell division. Right? Cytoplasm, kinesis, movement. So telophase is the end of what's going on with those chromosomes, getting them back into the nucleus and uncondensed. Cytokinesis is finishing up by separating the cytoplasm and the organelles and pinching those two cells apart. All right, so that is mitosis. Are there any questions over any bit of mitosis before we move on to talking about DNA synthesis? Yeah? So CNP, we covered CNPE, which is kinesin, and CNPA, I think, is the modified histone. Right, so that histone is what helps anchor the kinetochore structure to the chromosome. Right, it's interacting with binding with the DNA. CNPE, the kinesin, is anchoring the microtubule to the kinetochore. So one anchors kinetochore to chromosome, the other anchors microtubule to kinetochore. Any other questions about mitosis that aren't clear? Yeah. Sorry, say that again? You mean what directionality? The kinetochore itself, right? So specific proteins in it are directed towards plus or minus ends, but the kinetochore itself, I don't believe, has a charge. Any other questions? I forgot to look up the modified kinesin. Did you look it up? All right, I'll write it down for Monday. Questions? So it's simply the force is pulling on them from opposite directions, right? So you've got microtubules attached to each side of the centromere, and once they get the signal that the tension is right, they start reeling it in. So it's simply those microtubules as they're shrinking and the force pulling that apart. So it has to be really strong anchoring in the kinetochore, from the microtubule to the kinetochore to the chromosome to be able to pull that apart. Yeah? What do you mean by two motor proteins? You mean the kinesin and the dynein that we talked about? Right. Right. So the question is about, if you look at that drawing, and I didn't point it out, get back there. So the question is about the kinetochore. So there's a motor here, right? And then there's a motor here. So I didn't mention this motor here because it's not present in everything. So sometimes it's just depolymerization. Sometimes there's a motor there. But if there's a motor there reeling it in, what direction does it have to be oriented? Positive in, right? So they show it here, which helps, again, you're shortening and pulling from both ends to help really create the forces to pull apart those sister chromatids. But while there's always a kinetochore, this one is variable. Sometimes it's in, you get it in some species, and I'm not sure if it's animals or plants, but there's variation across biological diversity about whether there's a motor there or not. So that's why I didn't use it. But if it is there, it's got to be positive and directed, right? Good question. Any other questions? Yes?
So does that mean the one is the chromosome is
Has to be, right? Right, so it's pulling from both ends. bring in that sister chromatid.
But it's still a
It's a modified kinesin. And so that modified kinesin is still plus end directed, but there's no plus end really for it to walk down. So it's just serving as an anchor. Yeah. So it's already at the plus end. Any other questions about the motor? Good questions. All right. Oops, did that bit. Okay. So you have to know all those. You have to know the defining features of each phase. You have to be able to draw them, given any number of chromosomes that I think should be present. Make sure you pay attention to, if I say draw a particular phase of mitosis, how many chromosomes I want you to represent. Okay? All right, so we're going to move on then to DNA replication. We talked about cell division first, but in order for mitosis to happen, we first have to replicate that DNA. We also have to replicate the centrosome. We're only going to talk specifically about the process involved in DNA replication. We're going to go back and take a look at that S phase. Right? S phase for the synthesis phase. So this is where DNA is being duplicated, being replicated, being synthesized. Take your pick of verbs. All right, it's where we go from the G1 phase, one NTOC, one copy of all the chromosomes, to G2, where we got everything duplicated. So in order to talk about the way that DNA is replicated in the cell, we're going to start off by talking about how DNA is replicated in a test tube in the lab. I think in the lab this week everyone did PCRs, right? We did PCRs. So you should already be familiar with this. I think you did PCR also in the intro series. Right? So how do we replicate a small piece of DNA in a test tube? What's the process called? What is the specific process in the lab called?
Polymerase chain reaction.
Polymerase chain reaction, PCR. And this has become a standard, widely used technique in molecular biology. If you ever work in a molecular biology lab, you will do PCR. Sometimes even in ecology labs, right? Understanding what's going on at the genetic level is important. So we want to figure out how to get from 1 to 2 copies of a piece of DNA. So in order to replicate DNA, what is the minimum necessary requirement? We need an enzyme, a polymerase. We need DNTPs, primers, templates. Anything else? Need a buffer. Magnesium chloride, nucleotides.
DNA helicase?
What's that in the back?
DNA helicase?
Helicase, not in a test tube.
Oh, not a test tube, okay.
Heat. Yes. Right, so all these things are involved in DNA replication in a test tube. We'll get to helicase probably on Monday. All right, so at the very least, you need that enzyme, that polymerase. It's going to read your template and make a copy of it. And you need your DNTPs. Right, the building blocks to make that new copy. All those other things are important as well. They go into helping make these things work. What does DNTP stand for? Deoxynucleotide triphosphate. So it's a general term for those bases we're using as the building blocks. All right, what do we know about DNA polymerase? In order to build a new strand of DNA, what do we need? Where do we hook in the new nucleotide to?
Primer.
The primer. What specifically about the primer? Where do you attach that new nucleotide? 3' hydroxyl group. Right? So the polymerase requires a free site 3' OH for polymerization. It can't just start from scratch. It's got to have an end to begin off with. All right, so that polymerase, it adds DNPP to the new strand using a single-stranded DNA as a template. So it's not adding in the exact same base pair, it's adding in the complementary base pair. But it requires that 3' hydroxyl to begin. That's where that primer comes in. All right, I think someone already yelled this out in the back. How do we generate a template in the lab? I think back here somewhere. Here it is down front. Heat. All right, we looked at the melting kinetics, the melting temperature of a double strand of DNA. If we want to generate a single-stranded template in the lab, all we need to do is add heat. We heat it up enough, those hydrogen bonds are going to disassociate. The double strand is going to become single-stranded, and we now have a template. All right, so we have our polymerase, we have our template, we have our nucleotides. We still need that 3' hydroxyl group. Right, the polymerase needs that starting point. It can't just start from scratch. So that's where we bring in the primer, which is a short piece of DNA complementary to the part that we want to amplify. All right, now do we have everything we need? Yes, thank you. All right, so now the polymerase, it's got that 3' hydroxyl. It can start there. It can bring in a new nucleotide complementary to the template strand and begin to work its way down that strand, copying nucleotides as it goes. Bless you. So remember, right, when we add a new base to a growing strand of nucleic acid, we get the reaction, the condensation reaction. What kind of bond is this? Phosphodiester. All right, you should be able to recognize that. And make sure when you draw your nucleic acids for the exam that you get that phosphate in between your nucleotides in the right orientation. Right? All right. So this polymerase is going to work its way down the strand. So it runs out of template, and it's created a new complementary strand, and we're back to double-stranded DNA, and we've just made a copy of that. Okay, so for the exam, if I give you something like this, you should be able to tell me what the sequence of the new strand is going to be. Right? You can play the role of the polymerase, and you should be able to give me that strand the polymerase is making based on the template. Okay? I won't make you do this one. Anyone? 30 seconds? 5 seconds? How fast? You can have a race. All right, you just need to know your base pairing. A's with T's and B's with C's. I hear people racing. All right, so this is what, you should be able to do this for the exam. Give me the complementary strand based on any single strand piece of DNA that I give you. Also notice, I've indicated here, we talked about this format. This tells you what the 3' end is, right? So you know the directionality of these strands. All right, let's see if this video is going to work. Nope. All right, you'll also have to watch this one on your own. Basically, it's just a video describing the process of PCR. I'll put up a few more links on Facebook as well. There's some really great PCR videos on YouTube with, you know, like rocking dance music behind it that has nothing to do with PCR but makes it entertaining. So I'll give you a couple of versions of PCR videos to watch. If you're not feeling comfortable with PCR, please watch those so that you're familiar with the process. All right, a few clicker questions. Make sure you understand the directionality of what's happening in PCR. So I think you get the process, but things are happening in a very specific directionality relative to the DNA strands. So the first question, what direction does the polymerase move along the template strand? So I just pointed out the direction. I reminded you of how I'm indicating the directionality on the ends of the strands. All right, so this one is specifically talking about the movement of the polymerase.
That's the thing that adds the stuff.
Those are two different things.
Yep. It's the main thing that
This is specifically relative to the template, right? So you need to keep track of what I'm asking in the question, what the directionality is relative to. All right, everyone get a chance to answer? All right, let's take a look. Oh, split decision. All right, so the question, again, what direction does the polymerase move along the template strand? Oh. Didn't realize what the answer was? All right, what do you guys think the answer is? Right, B.
it adds 5 to 3.
All right, if we look at the template strand, 3' in, 5' in, the polymerase is starting here and moving this way along the template strand. All right? You need to keep track of what I'm asking it's relative to in the question. All right, so the polymerase is moving 3' to 5' along that template strand. All right, let's try the next one. The new strand is synthesized in what direction? All right, what am I asking? What's it relative to? Which strand are we talking about? That's right in that. All right, did everyone get a chance to answer? All right, let's take a look at what you think. All right, better. All right, it is 5' to 3'. Right? We're synthesizing, starting here and moving in that direction. 5' to 3'. All right, I think I have one more before I let you go for the day. All right, this one's a trick question. DNA polymerizes DNA from the what to the what? So what is polymerization? exactly is polymerization. Right, so you have to understand that to understand the question.
So it's adding 5 to it's taking polymers and adding to polymers.
But I was thinking like because you look at the relative difference in
Everyone get their answers in?
But it's not connected to the right?
Take a look at what you think.
okay, no, that's right.
Don't ask me. Not second guess.
Anyone want to justify their answer? Say that again? So the answer is, right, so it adds to the 3 prime. So polymerization is the process of reacting those monomers, those nucleotides together. It's the condensation reaction. What does polymerase need to be free? The 3' OH. So the polymerization happens from that 3' OH to the 5' phosphate. All right? All right, I'm specifically talking about the polymerization, that condensation reaction between the nucleotides. Start with that 3' OH, bring in the 5' phosphate. Good question. I warned you. All right.